Good morning, Professor Sutipong and dear friends and colleagues. My name is Dr. Prapan Panupak. I used to be the HIV-NAT co-director. Now I'm just the advisor of HIV-NAT. I would like to welcome all of you to this 26th Bangkok Symposium in HIV medicine. I know that many of you are still the so-called frequent attendance of this meeting, so I am very grateful for all of you here. So it's my honor and my pleasure to introduce the first speaker today, Professor Kiat Ruxrungham. Many of you know him as a famous HIV clinician. He's now working on HIV, non-HIV, COVID vaccine, especially on messenger RNA COVID vaccine. He's now the director of School of Global Health, Chulalongkorn University, as well as the co-director of the vaccine program at the medical school here. Professor Kiat is going to give a talk on messenger RNA vaccine and its development here. So Professor Kiat, please.
Thank you very much, Ajahn Papan, for your kind introduction, and my great honor to join once again HIV-NAT Bangkok Symposium. But the topic I'm going to focus is gearing to another direction that in the past 4 years or so that our Vaccine Research Center has been developing. So the topic is the mRNA vaccine development, the pandemic preparedness, and beyond. If you look at the history of pandemics in the past century, you can see that in the past century, the pandemic's interval is, in average, is longer than 20 years or more. But just recently, in the last decade, you start to see the interval between each pandemic is much less than 5 years. And we believe that this phenomenon getting more and more serious. I just checked, if you check the WHO data on the COVID pandemics, and you can see that this is official or confirmed report of the cases. And all of you realize that particularly the total confirmed case is seriously underreported, because many of you, including myself, recently you also got COVID and you didn't report at all. But anyhow, the confirmed number is greater than 700 or up to 800 million cases, and more than 7 million cases die from COVID. We have been gone through a very serious impact of pandemic by COVID-19 in the past 4 years, and in various aspects, including health, life, economics, and society. But one extraordinary phenomenon in science is about the vaccine development. And it's never before in the world history is that developing vaccine against one pathogen is more than 240 candidates. And more than 800 clinical trials has been conducted related to COVID vaccine, and more than 80 countries involving. And at least 50 COVID vaccine had been approved, either at the national level or some quite a number at the global level. And you see that this very positive impact on the vaccine science in terms of speed of the development, number and types of the vaccine technology, efficacy, and the rolling out implementation. However, we're still seeing a problem of inequity on accessing the vaccine. Now, this slide is just briefly to give you the idea that what the vaccine technology platform has been used in the last 4 years during the pandemics. On your left-hand side, consider as a new technology because it hasn't been approved for human use before COVID. And on the right-hand panel, just a second. Oh, sorry, I don't think we have a pointer here. Okay, all right. You see that this is the earliest existing technology vaccine of COVID is inactivated vaccine, and followed by viral vector and new technology, and mRNA, and recently is a protein subunit vaccine. Now, when you look at the timeline of developing a vaccine through the approval for clinical use, in the past it takes much longer, and some of the target infectious disease still we cannot have approved vaccine available. But just I give you a few examples in the last 5 decades. You can see that like Ebola, it takes 43 years, hepatitis B is 16 years, and HPV is about 25 years. But with COVID vaccine, it's been taken only less than a year. So what makes that a difference? There's 2 or 3 factors that contributing. The first thing is the advanced technology plus the well-settled existing technology like inactivated vaccine. But more importantly, also because of the incidence of the disease, transmission is very aggressive. If you remember, 2020, we shut down, even shutting down, you're still seeing people got serious, severe COVID disease, and elder people particularly dying. So because of the incident and spreading is so quick, you can conduct the phase 1, 2, and efficacy trial with less than 6 months or 9 months. So that's why the vaccine got approval with less than a year because of the efficacy of the vaccine as well. Now, if you look at the data by Google and say that how many vaccination doses of COVID vaccine had been given globally, this is the updated data from WHO. So it's more than, or it's up to 14 billion doses has been given worldwide. And you all realize that the booster vaccine so far globally, including Thailand and anywhere, mainly is mRNA. So if you look at, let's say, the assumption of about 40% of those 14 billion doses given to people are mRNA vaccine, that will be at least 5 billion doses of mRNA vaccine has been given. But if you look at the map, you can see that the vaccine inequity issues remain. So particularly in the sub-Saharan African continent, you can see that a number of area of country still has less than maybe 40% of people receive just only first dose. Now, the question is why the mRNA vaccine has been developed. Is that because by chance that got the mRNA within 4 years? So I'd like to highlight the history of mRNA vaccine development. Actually, scientists has been discovered and named as a messenger RNA. Actually, before 60 years ago, a number of scientists have been working on a molecule supposed to, that is a mRNA, but until 1961 has named that as mRNA, a messenger RNA. So, and until 40 years later, this mRNA has been discovered that if you modify the mRNA sequence, particularly the viral RNA sequence, and giving to human, you can minimize innate inflammatory responses. And the other, by 2005, also lipid nanoparticle scalability is feasible. And 7 years later, the first mRNA vaccine encapsulated with lipid nanoparticle has been tested in mice. And 1 year later, the first vaccine is not COVID, it's rabies mRNA vaccine get into human trial. And the lipid nanoparticle, the first one that has been licensure for clinical use in the US, actually is not mRNA vaccine, it's the siRNA. And the name of the product is a patisiran. So this is a siRNA to silence some of the protein to treat a rare hereditary polyneuropathy disease. So you see that a number of knowledge and technology has been accumulated before COVID hit the globe. And when 2020, actually we learned that COVID started at the end of the year 2019. That's why we call COVID-19. But it's become pandemics 2020. And at least two biotech company, BioNTech and Moderna, start the vaccine trial and get approval within less than a year. Now, I think mRNA vaccine is one of the good example of human or scientists learning from nature. So nature is really a super amazing. Every of us, if you search at Google that how many vaccine you can be produced by one individual, is about 20,000 proteins. You have gene, you have chromosome, have gene, but the coding genes, DNA cannot make protein directly. You need to make a short-lived messenger RNA. So it's a very short-lived and make protein, specific protein for you. So in this case, the mRNA is short-lived, is not integrated into the nucleus that people are carrying. And this short-lived, when is produced specific protein that you want to produce, then it will be degraded. Now, there's a number of scientific evidence, and this is quoted by the International Infectious Disease Society in the US. And you see that the misinformation of mRNA will integrate into the nucleus and create a lot of trouble, cancer, and autoimmune disease is misinformation. And I like to emphasize again that it will be degraded within days. So it means when you design a vaccine to make spike protein, it will, one is mRNA make protein in the ribosome and make protein of spike. For a few days, mRNA will be degraded, and that protein will act as the antigen to stimulate your immune response to recognize the pathogen that's going to invade your body in later on. Now, the question is why mRNA vaccine has been taking so long to become able to be clinical use. That is more than 60 years. There's two obstacles. One is mRNA sequence, particularly the viral mRNA RNA sequence, will strongly stimulate innate immunity. That will generate a lot of side effects and reaction. Secondly, mRNA is very, as I said, very fragile, so short-lived. So you have to cover it and deliver it into the cell without degrading before making protein. Now, how to avoid and reduce innate immune reaction to mRNA, particularly the viral sequence to make viral protein? When you get viral infection, whatever, common cold, RSV, flu, HIV, whatever, you generally in your cell, at the cell level, you have a lot of what we call the viral RNA sensor. So you look at the cartoon, so I don't think we have to go into the detail, but a lot of sensor that can trigger your body to alarm yourself that something, particular virus invading your body. So you're going to have fever, fatigue, anorexia, and some other specific target involvement as well. Now, this is the discovery by Professor Drew Weissman and Kathleen Calico. This slide courtesy from Drew. And you can see that when they look at the monocyte-derived dendritic cell, and so highlight in here, if you use the viral sequence RNA without modification at all, you generate a strong TNF-alpha. This is very strong pro-inflammatory cytokine by the monocyte-derived dendritic cell. In contrast, if you switch the native uridine into modified pseudo-uridine, and then you shut off all the inflammation that are related to side effects. The second obstacle to overcome for making mRNA is how to deliver it to the cell without a rapid degradation. So this is a discovery and scalability of lipid nanoparticle. So lipid nanoparticle composed of 4 lipid components. The first one is the cationic ionizable lipid, cholesterol, helper lipid, and stabilizer. In this case, is pegylated lipid. Now, with that discovery, Prince Mahidol Award, and about to have the Prince Mahidol Award conference this year, actually this month. So in 2021, have award an honor to these 3 scientists. So 2 are related on the modified nucleoside to reduce toxicity of mRNA. And the last one is Professor Peter Cullis from Canada is related to lipid nanoparticle. And just this year, 2 scientists has been awarded by the Nobel Prize. So Kathy and Drew, both of them, related to modify mRNA that reduce inflammation reaction that make vaccine can be usable in human. Now, what are the pros and cons of mRNA vaccine? The first thing is, and I can show you it's real in our lab right now, can be rapidly designed and developed very quickly in weeks. And you can make the prototype in less than a month and go to animal testing within less than 2 months. So this is really robust technology. Second thing is the induce strong neutralizing antibody and strong Th1 response. Scalability is also a beauty of a pros of mRNA vaccine. Right now, it's very clear that you can use a very small footprint for less than 40 square meter to make about 1 million doses of mRNA. So the technology is very helpful in terms of for future vaccine development and pandemic preparedness. And it has been proven the highest vaccine efficacy against COVID-19 compared to the conventional vaccine technology. Potentially, in the near future, will be low cost in long term because raw material now be able to make in Asia in Korea, in China, and well in the future will be in India, can be also used as a personalized therapeutic vaccine for cancer and other chronic diseases. Now the con is that because this is a messenger RNA to make protein, if you want to work with any antigen that not protein, like polysaccharide, like pneumococcal vaccine, you cannot use mRNA. And it needs a low temperature cold chain. But right now, it's very clear that some formulation can stay at 2 to 8 degrees up to 6 months. But if you lyophilize the vaccine, it can stay at a 2 to 8 degrees for years. And the concern is still there in terms of rare myocarditis concern in young male. And the average, the incidence is about 10 to 20 of a million injection dose. Now, at the Chula Vaccine Research Center, we have found it a decade ago and have been working on DNA platform and subunit protein platform for quite some time until a few years before COVID started. We believe that mRNA vaccine is the new disruptive technology. So we start inviting Professor Drew Weissman and collaborating from that time, several years ago. So our goal is to make the mRNA vaccine more accessible in low and middle income countries. And this is the commitment between Drew and us that we really mean to bring it to be more accessible. So after we got the knowledge and tech transfer from Drew, now we be able to more independent that can develop a vaccine very fast and high quality. The first design vaccine we call COP19 is the first generation. When we develop, we test in animal, it's beautifully immunogenic and toxicity study also shows safe, and then we move to GMP. By the time that our GMP facility in Thailand is not available yet because they have to set up, so we use the CDMO in the US to make the first clinical lot for us to prove the concept to the government that is workable. And the data of phase 1 this is data in animal study. It may be too small for you to see it, but it published in Nature Communication and showing that the vaccine very immunogenic and cross-neutralize again a number of variants, but less against the Omicron. And then the phase 1 data also published in Nature Microbiology and showing that Chula COP19 mRNA vaccine is a dose-dependent fashion in young adult and elderly. But you can see that, and many of you, and including myself, that consider as an elder, always have a much lower immune response compared to younger adults. And then we pursue to phase 2. It's a randomized 4-to-1 study. So 120 receive Chula COP19 and the other 30 receive placebo. And that time, the placebo we given for 1 month, and the delta, data is picking up the delta variants. So the SMB recommend us to switch all the placebo after 1 month to Pfizer vaccine. So this is the data. As you can see that in blue is the neutralizing antibody generated by Chula COP19 and the placebo group, and then they all receive community is the Pfizer monovalent vaccine, and you see that the result is very promising. Now, based on that phase one data in particular, we'll be able to convince the Prime Minister to visit the lab in a 2 day later when we announce to the public and fund us more money to pursue production in Thailand and pursue further clinical development. And this is just a brief summary of the clinical development, the Thai-made first generation vaccine monovalent. Again, when you develop something new, every sector that involving always have the lesson learned to take. Our regulatory reviewing process has been taking quite a long time to make a decision to allow us to start phase one. So so that's why we decided to start a parallel trial in Australia. And then when we start the phase one Thai-made vaccine and in Australia, the global Omicron outbreak is starting, including in Thailand. So it's no reason to keep pursuing monovalence. So this is proven that we can switch to design the bivalent in the lab and test in animal very quickly and start making the vaccine. This is made in BioNet Asia. It's a Thai-French manufacturer at Ayutthaya. So in this time, the Thai regulator take shorter reviewing process, only 4 months, and we start phase two in Thailand, 450 volunteer and also in Australia. Now this is the dashboard showing that in clinical trial, Thailand, we are very efficient to do it in a very high quality. So less than 2 months, we can enroll and complete the enrollment of 450 in 4 sites within less than 4 months. So it finished by the end of November last year. And in Australia, still missing another 5 individuals. So hopefully in next week or so, we will finish up 2 sites, two countries trial. Now, with our capability right now at the commitment that we want to be part of the contributor for vaccine equity, so we join the global network. So in the Thai setting, it's not really only one player important. So Chula VRC, involving in the vaccine design, animal testing, and prove that it's a good candidate, and then move to and collaborate with VRC at Penn with Professor Drew Weissman and funding agency. The good news is that during the pandemics, National Vaccine Institute now become one of the key grantor funded by the government to support vaccine development and working with the vaccine biotech in Canada and in Thailand. And now we join the WHO mRNA Vaccine Hub R&D network. We focus in our Asian region, and we in part of reading some of the target vaccine. I'm going to share with you in the next few minutes. Now, that the pandemic that we are facing, now what about mRNA vaccine being developed beyond COVID-19? It's quite a number of viral target had been working on and can be searchable in the literature at clinicaltrial.gov. But what I'm going to highlight is that, in this slide, you can see that 2 viral target vaccine entering or about to finish phase 3. So it's a RSV by Moderna, and they're looking for the final approval, and the influenza vaccine also by two biotech, the big mRNA vaccine player. So phase 3, 6,000 individual run by Moderna, and 25,000 run by Pfizer-BioNTech. And they're also about to, in the process of getting reviewing process for approval. Other mRNA vaccines like Zika, rabies, malaria, PF, and HIV still in a very early clinical development. There's one more phase 3 viral vaccine mRNA is CMV. Now, let's focus on our center, Chula Vaccine Research Center beyond COVID. So the pandemic part I have been presented, and we start joining with WHO and Global Network and to prepare for the next pandemic. Now, with the newer facility, our either Chula-initiated vaccine, and we be able to produce R&D vaccine for other collaborators, both in Thailand, in outside Thailand, and also through the WR&D network. Now, we can make antigens mRNA encapsulated about 10 antigen per month. With the new facility that will be able to start operating by the end of the year, we should be able to increase to up to 30 antigens per month for the R&D. Now, this is the list of the target vaccine that our center either initiated or collaborating. You see that. The one that I highlight in bold is the one that had been tested in animals. So dengue tetravalent therapeutic HPV vaccine. This is a PYVAC, is led by Ajahn Yesumon from Tropical Medicine, Mahidol. And we also have How Does My vaccine. So what I'm going to share with you in brief, at least 2 vaccines that we have very promising data. So one, we all realize that HPV is a challenging problem globally. So right now, the world population is about 8 billion people living. And you look at now in terms of preventive vaccine, it's great. It's now the viral live particle vaccine, HPV, is wonderful. It's the best efficacy, up to or more than 90% efficacy. But the problem is the accessibility, and people everywhere, internationally and the national level, try to enhance the coverage. You see that the data 2-3 years ago, the coverage is only 15%. Now, when we look at why we are interested in therapeutic vaccine, because the data are showing that up to 300 million women infected with HPV, so it means DNA positive, and about 1/5 of men, this is some of the cohort, infected with HPV. So the preventive vaccine won't work, and you have to just, hopefully they're going to cell recovery, or in the next 10 or 15 years will become precancerous and cancer. So what the rationale of therapeutic vaccine? There's in the last 10 years or so, there are quite a number of attempts to develop therapeutic vaccine. When we're talking about therapeutic vaccine, you see that the first thing is if we can target the precancerous state and bring the sin tree before become cancer regress to be normal, that's the goal. The second goal is to get rid of the DNA. And the third goal, which is tough, we learned that if you have a big solid tumor, forget it, that vaccine alone will work. You have to work with it's an adjunct therapy mainly. So our goal is, number one, is to make regression of the early tumor or precancerous and get rid of DNA. Now, this is very, very preliminary data, and we have a lab meeting yesterday, and data is very impressive. So none of this data has been published yet, but I think it's worthwhile to share that this is maybe a promising candidate to pursue. You see that this we work with the E2, E6, E7 as a target because in the last decade has been proven some of it or maybe combined of this antigen can reduce the tumor size very significantly. So we start, you look at the protein expression beautifully, very beautifully. And when you immunize mice with a low dose, 1 microgram, you see a good ELISpot gamma interferon response. E2 is higher than E7, and the lowest one is E6, but still better than the control. And this is the CD8, because if you work with infected cell or early tumor cell, you need strong CD8 response. And this is the gamma B positive CD8 T cell. All three antigen induce a good CD8 response, either expressing or producing gamma B or TNF alpha. Now, and we're very lucky that we have good people in the team. This lady, Supisha, our postdoc, she can set up a tumor model very quickly by collaborating with a number of labs to get the cell line. So this is a T1 cell line that generate HBV-related cancer. So you look at the control is the normal cell line group. You see that day 8, you start to see a tiny spot. But day 11, the tumor getting bigger, 13, and 17 is too big. And by this animal safety criteria, if the tumor size greater than 1.5 centimeter, we should terminate or take down the animal. So the animal in this control arm has been taken down. And you look at the vaccinated group in this in E6, E7, you see the small tumor size is still smaller, particularly the E7. And the way to look at the result, you can see that in red is the control arm, day 7, and the green is this is day 17 of the control arm, and this is vaccinated but E7 and E6. You see the tumor regression time rate is about 34 smaller, 11-fold smaller. And keep in mind this is we use 50,000 cells. So we now start to, because the goal is the early cancer, so we start with 10,000 cell transplantation, and we see the result in the next few months. Now let I switch the gear to another candidate vaccine that my background actually also not only immunologist, it's also allergist. So we still have a passion to work on some of the allergy vaccines. So in this case, you see that allergy is still a problem. I believe many of you here, at least 1 of 3, sneezing, running nose, and 1 of 10 may have asthma, or 1 of 20. So it's a common diseases, and the most common cause of allergy in tropical area in particular is house dust mite. And it has been proven and licensed for clinical use for decades is the immunotherapy. So either by subcutaneous skin or sublingual. But the limitation is that the adherence is low, and sublingual even lower, because of you have to take it almost every day. So the question can be develop a technology that less immunization needed and a strong Th1 response and Treg. Now, this is the target profile design of the vaccine. So the goal of making candidate vaccine, we should aim to increase IgG4 blocking antibody, induce specific Th1, induce Tregs, and suppress IgE production. And when you challenge the animal sensitized with haldosmide, you should be able to block eosinophil inflammation in the airway. Sorry, the slide is distorting, but you can see maybe a bit difficult. So this is the control, producing IL-5, and this is all different immunogen design, so we don't need to go into detail which immunogen. We're selecting the best one. And you see that is the target goal of making a candidate is to inhibit IL-5 production. You look at some candidate can inhibit 90% or more of IL-5, and you can expect that eosinophil production will be dropped. And looking at the regulatory cells, T helper cell in the follicular T helper cell, IL-10 production and neurotin production, you see this is a control. Some of the candidates produce a strong one. And I'm really sorry that it needs to come out. So you have to believe me now. So in red, actually, okay. So this is a bronchial alveolar lavage, and the red color has disappear here. It should be like here. But look at this inhibition, reduction of eosinophil. So again, different antigen design can suppress eosinophil response after challenge in animal very strongly, and they also suppress IgE production. So this candidate already funded by the income of the university. We call the Chula Second Century Fund to allow us to pursue for phase 1 clinical trial. Now let me end by again to remind all of us that pandemic is not too far away. It's coming sooner and sooner in terms of interval. The best way is should not rely on only 2 or 3 big vaccine company, but to this is the attempt of WHO and so on, CEPI and other, that each region should set up the capacity and be able to prepare all the prayer to make sure that when the next pandemic started, we can generate the prototype, testing preclinically, and manufacturing is high quality, and do clinical trial very quickly, and not as country-based, but should be a public good vaccine approach to a regional or global network with speed and quality. So to do that, we believe that the mentality of thinking should not competing between countries. We should the WHO consortium right now start to see that type of phenomenon that countries in Asia working together more and more, and joint funding and so on and so forth. So we strongly believe that vaccine equity is feasible because of this disruptive technology and the capacity building and the more meaningful collaboration of regional level and global level. I'd like to thank all the collaborators both in Thailand, outside Thailand, in public and private sector, clinical trial staff, and and also the Australian company Technovaria to coordinate 5 sites trial in Australia, and the funding. I'd like to emphasize that we got a strong support from the government, but at the same time that we are able to be sustainable is by the donation. So by those donated money, we are now expanded our capacity and can do more work. Thanks to the audience, thanks to the volunteer, and thanks again to my team. Thank you very much.